Well, you've mentioned the lab a couple of times. It's a few steps away from us. Would you be up for us having a quick tour of it?
Let's do it.
Okay, let's make it happen.
All right, so here we are at Eigen Therapeutics wet lab. Transon, would you like to give us a high-level overview, and then we can dive deeper into each of the different sections of the lab?
Yeah, sounds good. So welcome to our lab. For those of you that have seen biolabs, some of you may be used to a lot of kind of fixed furniture in place. We do have some fixed furniture, as you'll see some of these classic lab shelves that are up against the wall. But one thing that we really like about this lab is that there isn't really a lot of fixed space, right? When we move in, as you sort of start to look at all of these tables that are here, everything's movable. It's essentially one big open space. And so if you want to build out a lab in the way that you want, that kind of integrates both automation and bench work, that's great because you've got a big old open canvas. You can lay things out the way that you want. So you'll see as we start walking through the lab, there's some areas that are pretty automated, some areas that are semi-automated, and some areas that are pretty classic, really just bench work areas. And I think it's pretty important to lay it out that way because when you're doing R&D discovery, everything changes all of the time, and you can't really make a fully automated system for everything. So every place has to have their home, right?
Cool, yeah. So can you walk us through what are the different homes and maybe how has it evolved over time?
Yeah, for sure. So in this area, you'll see our liquid handler. So this is basically a 384 head. So you can imagine it's the equivalent of 384 pipettes in one head that'll move around. It's very good for stamping. And so we use this for any sort of large scale stamping or creation of our plates. We do a lot of drugging, for example, and so we have to create these compound plates that have a bunch of different drugs at different concentrations. And this will help a lot with that.
Cool. So this is walk-up automation used by the scientists directly.
That's correct. One theme you'll start to see here, aside from our integrated work cell over there, is this idea of kind of separated areas of automation, but still with a software layer on top of it to make it as easy as possible for scientists to use. So for example, with this machine, this is a little homemade app that we wrote ourselves with the little Eigen logo to just sort of choose a method that we've created. You tap one of these things and it'll just kick that off and start your method. And so I think there's a lot of promise here in building software on top of your automation to make it easier to use. And you don't really need a fancy robot arm or an integrated system to do that.
Cool. Yeah, one thing I love about Eigen Therapeutics Lab is being very practical about what you automate physically, and then you also have a software engineering background to automate some of the data steps.
Yes.
Is this an example of some custom software, or is this purely a Hamilton method configuration?
Oh, that's a good question. So on the back end, it is still Hamilton. So you click one of these buttons and it'll essentially call up the Hamilton Venus method in the background and start it. This is just a micro app that solves the very simple problem of where's my method and how do I run it? And that's really important too, because one part of our engineering philosophy here in the lab is working on, so to speak, the boring stuff, right?
Boring stuff, yeah.
If you work in a lab, there's so many little micro frustrations that happen. Shoot, where's the pipette? Oh, I need to start the stamping method. Where is it? Is it in C/programs? Is it V2, V3? I think just taking the time to work on all these little boring issues really add up. It just, it's more like you won't even notice how smooth it is. Otherwise, highly complex labs with lots of things going on can kind of slow to a halt, not from any one thing, but really just from death by 1,000 cuts. And so that is another common theme we'll see here. Micro apps here that just solve the problem of help me start a method.
Exactly, yeah. Cool.
Other general bench space here. So as I've mentioned, we do a lot of high throughput flow cytometry. And so this is one of the flow cytometers that we use. Works with 384 well plates. Most of our screens are at a 384 well scale. And so for those who, if you're familiar with flow cytometry, it works very similar to a flow cytometer. There's a probe that will aspirate cells and then look at surface expression of targets after you stain them. This is a probe on a gantry that will do that for every well in a 384 well plate. And again, What you don't see here is all the software behind it. If you are reading a lot of different targets, you have a large panel and you're getting a lot of stuff out of every single cell, out of every well in your 3D4 well plate. It's a lot of data. And the act of starting to even get that data off of this computer and into the cloud in a way that we can analyze it starts becoming really important. So all of these little instruments that we have that are kind of isolated on their own still have a layer that makes it really easy to extract all the data that comes out of them, put it into our cloud, start analyzing it.
Into your own custom-built software suite you call Vector, is that right?
Correct, yeah. So Vector is our LIMS, our Laboratory Information Management System, completely built by ourselves. I can give a brief overview of it in a second too. Yeah.
I was curious, what's the benefit of building one yourself versus taking something that's available on the market?
That is a very good question. And I will say right now, as an engineer myself, and as a company that has decided to build our own LIMS, that not everyone has to build our own LIMS. We did build it ourselves because we had the capability to. We knew what we wanted to build. We knew how we built, how we could build it. And for us specifically, it was building it exactly the way that we wanted, customized just for us. And that also means, we think about science in a particular way. The schemas, the sort of columns that we'll have for certain tables kind of map to how we naturally think about our experiments. And so we built our LIMS perfectly for the way that we, as scientists, will think about our science. So it's great for Eigen. If you were to take our LIMS and give it to another company, it would probably be terrible just because they think about science. Most companies think about science in slightly different ways.
As you talked about the modularity of your lab. Is there a similar sort of framework that you're using in your LIMS as you plug in new equipment, for example?
Yeah, that's an interesting question. I think there's always a bit of play between sort of modularity and ease of use. Engineers, and I can say this because I'm an engineer myself, I think we're always guilty of trying to put things in a nice, clean little box. And if you're doing science, especially R&D discovery where stuff changes all the time, that's just really hard. You're going to try and learn every single step that everyone ever does, and you'll try and create that in your system and give them a menu of items that you can build, and that's perfect. And then tomorrow, someone's going to want to do something that's outside of your system. And that's okay. They should do that. That's how innovation works. So your system will be broken over and over and over. And so it's really just finding that balance. Sure, find the things that you do a lot, maybe productionize them, try and build into a system, turn into a menu of items that you can sort of make into a modular workflow. For example, our main screen, to highly simplify it, is do a lot of drugging to a lot of cells and then read them out in flow cytometry. And some of those bits we'll do over and over again. And so that makes it pretty modular. Scientists can put together these blocks. I'm going to screen these drugs against these cell lines at these concentrations. And the software makes it really easy. At the same time, scientists will always break that workflow because they should. It's how you do new things. And so you wouldn't necessarily want to support that with strict engineering. You just want to give them the means to do so while still capturing what they've done in a loose format, or at least getting the data.
Yeah, so some modular building blocks that you put on wheels so you can reconfigure things.
Yeah. So as we start talking a little more about integration, this is the area that is a little more automated. So this would be our integrated work cell. You've got most of the time when people think about robotics and automation, they think about the robot arm. It's kind of the classic idea of automation. I've got a robot moving plates around. I mean, the robot arm is cool, but what I'm really interested in is all these different instruments that actually themselves do a very important job in the process. The robot arm is really the broker, right? Whose job is to carry plates from instrument to instrument as each instrument does some kind of perturbation. So this work cell that we see here automates some of our core processes. There's compound addition that the process I mentioned before of drugging your cells at high throughput, as well as a lot of the steps for flow cytometry.
I see. Yeah, I see you have another flow cytometry here on a similar cart with wheels.
That's right. Yep. So this one is the one that's closer to our automated work cell. And in flow cytometry, there's a lot of standard steps that you would do on a benchtop that you pretty much want to translate into an automated system. When you prepare cells for flow, you want to block it. You want to stain it, do some various wash steps, and you load into your flow cytometer. You basically just want to replicate all of those steps, but in a 3D4 well format through robotics. And that's basically what we've done here.
Very cool, yeah. So you do staining on here as well?
That is an example of semi-automation. And so we will actually use some of our other robots to do staining.
Got it.
And then have people move them to a different portion of our lab.
Got it,
Yep.
yeah.
So that's where you can have like little islands of automation. This little automated area does steps 1 through 6. You move it to this other automated area that'll do steps 7 through 10.
Makes sense, yeah. Yeah. So just to recap, this island of automation helps you create compound plates, maybe do some staining. This section helps you do readouts. I see a citation 5 there in the flow cytometry. And then this island kind of puts everything together, but does it at high throughput.
Handles the core parts of drugging and flow cytometry.
Drugging through flow cytometry, yep.
And we can even make the robots move a little bit. We have a typical test suite that we'll run. Just like in software engineering, you want test suites for hardware as well, just to make sure that all of your teach points are correct for your arm. So I can go ahead and kick off something like that. What you're going to see here is our robot arm is going to pick up this test plate that we have with a lid, and then just move it through some of our most common motions.
Oh, you got a mini sense. Cool. I'm a big fan. That's awesome.
Yeah,
Yeah,
they're great. Yeah.
Cool. I didn't realize you got one since I last came to visit.
So now it's placing that plate into the Echo. The Echo is a pretty awesome low volume liquid dispenser. It'll shoot 2.5 nanoliter droplets. And so that is our workhorse for compound addition. We're basically shooting nanoliter scale droplets of various compounds into the cancer cells.
Cool. Yeah, do you want to walk us through the different functions of each instruments, maybe?
Yeah, sure. We can do this live. So we're going to see this arm pick up that Echo over there. And then actually, if we move over to this area, you'll get a better view of the next few steps. A common step in washing is centrifugation. And so we're going to see this arm pick up that plate and then put it into our automated centrifuge. Again, this is our test suite, and so you're not really going to see the centrifuge run. It's more making sure that the arm can actually pick and place plates from the proper positions. A common step that you'll see after that would be shaking. And so this is a plate shaker, and we'll see this arm move over here, place it on that shaker. And we'll see the locks close, and we'll see it shake for a little bit.
How sensitive is this process to, moving, for example, a table by an inch?
An inch would be pretty catastrophic. Yeah. I'll usually say 1 millimeter is where you start running into problems.
Wow.
That said, if you can design your automated systems such that they're pretty robust to changes like this. It's pretty tough to see, but these tables are pretty heavy. They're all bracketed to each other. I have pretty strong confidence I can just come right here and kick this while it's moving, and everything will be fine. That said, you never know when things will change, and that's why, just like you always run test suites in software, you're going to have test suites on hardware too to make sure nothing is drifted. And so this is our limbs vector. Again, it was completely made in-house. Back end is Ruby on Rails. Front end is Next.js. Really handy thing. It's all built on a GraphQL API, and so that also makes it a lot easier for some of our AI agents to kind of just discover what kind of APIs are available and automatically search through stuff so humans can ask natural language questions and get structured answers back from our limbs. But we've talked a bit about modularity. This is an example of one of the screens that we run. You kind of see these little blocks that allow scientists to put together their tasks. I'm going to plate cells, and I'm going to drug these cells on day 0. And then if I click on these plates, I'll be able to see what drugs that we've had and where each compound is going to go. So this one's a pretty simple 2-plate screen. I can go over here and see all the different drugs that we have used on this assay plate. And again, at this scale, I'm not going to remember what drug went into what well, and this is where you're going to need the software to not only help you put together the data, but to even just plan where you want these drugs to go. So I can click on anyone, let's just say well E10, and I happen to know that's one of our controls. We're shooting DMSO. But I'll also see what kind of panel we've used when we've stains. But data like this is really important. Ideally, you do all this planning, as much of this planning as you can from your desk, such that you can then walk into the lab, and then all this information that you've planned funnels down from your limbs into the robotics that your robotics executes.
I'm curious what the learning curve is for scientists as they come into the lab and they're trying to interact with this limbs. How do you manage that?
Yeah, I will say this is one of the benefits of building in-house, and going, a callback on how important empathy is. Your coworkers, your users or customers, they're right there. It's your coworkers, it's your scientists. So, when you bring in a new person, you teach them a little bit about how to use the limbs, you watch them, you strongly encourage them to come to you with any questions, feedback, and you get a sense for what works and what doesn't. And if it looks like there's a part of the limbs that's a little hard to understand, or if there's just way too much clicking going on and it's starting to become one of those things that eventually leads to death by 1,000 cuts, you can fix that yourself. You can find a way to streamline it. You can hard code various steps to skip over that because you know that that works for that particular workflow. So that's a benefit of just making it in-house and just making sure you have that empathy with your users who are right there next to you.
Absolutely.
Yeah, you mentioned death by 1,000 cuts. Specifically, this view here. What's the cut that you've resolved? Is that like walking back all the way to your desk to look up some data?
Yeah, I mean, the first cut that I think is the most prevalent to me is this sense of what did I do with this screen? Yeah, when you've got a few conditions or a dose response, and you're trying to do it all manually, you'd probably structure it in such a way that you know exactly what conditions you've done in each well. There probably aren't that many conditions. But again, when we're talking about this many conditions, you simply just can't fit it into your brain. You can kind of see from some of the colors here that we're randomizing it as well. And so I definitely won't know what went where. And so using this to just do all of your planning first is a major cut, or at least addresses one of those cuts.
Do you have any favorite instruments?
I'll give two answers, and they're not really focused around a particular vendor. But for us, I mentioned this one instrument, the Echo, which is the nanoliter dispense machine, and it's particularly helpful if you're doing something like what we're doing, a lot of different drugging of cells in a randomized format, and we want to use a bunch of small volumes. Depending on your use, you could work just as well with something like that machine, which is essentially an automated pipetter, which would work anywhere from, the microliter to mil stage. But machines that I really like are actually something that's a little simpler than that. You've got these machines that kind of live at the intersection of kind of automated and kind of not. Exactly. This is a 96, actually this one's a 384 head. They're swappable, so I have to actually check. And this actually can be programmable, and we have programmed it to be able to move around and dispense for us. But again, in an innovative setting where scientists are doing new things every day, you can't hard code all of your protocols into your liquid handler and say, here's your menu, here are the things you can do. You want to give scientists the freedom to just say, I've got this cool idea. How do I use these tools myself so that I can just test out my idea right now? And little instruments like these that kind of live right at that intersection are super helpful. Because sure, you've got a 384 head, but you can also kind of just move it around yourself if you want, and kind of just manually do your pipetting.
Cool, yeah, I often get asked, should we automate the lab or not? And it's this binary thing, but it's like a whole spectrum of options.
It's a spectrum, and I think you got to make room for every space in the spectrum, from fully automated, pretty locked down, to highly exploratory and changing every day.
Well, I'm hearing a common theme of just reducing friction from when a scientist wants to do something to actually being able to execute it. And
Absolutely.
how you design this.
Yeah.
Really cool.
Reducing friction is extremely important, and it goes back to that same thing of stopping death by 1,000 cuts. If you just have something that you dream of, how can you design a lab with all the tools and infrastructure that scientists can just execute on that dream?
Very cool.
Cool, yeah.
This is great.
Thanks for showing us like the whole spectrum in one lab. That's super cool to see. Yeah.
Of course.
Awesome.